Imagine your gut as a bustling city, where trillions of microbes act like workers, breaking down food and producing tiny molecules that power cells, signal the immune system, and even talk to your brain. In this lesson, we Eurostrade explore how these microbial processes keep your digestive tract healthy. The human gut contains a vast community of microorganisms that outnumber our own cells by an order of magnitude. These microbes are organized into distinct groups, or phyla, that dominate different regions of the intestine. In the colon, two major bacterial phyla, Euros firmicutes and Bacteroidetes Euros, constitute roughly 90% of the microbial population. A smaller but important third group, Actinobacteria, contributes to vitamin synthesis and mucosal protection. Each bacterium interacts directly with the intestinal lining through surface proteins that bind to epithelial cells. These interactions trigger signaling pathways that regulate cell turnover and barrier integrity. The microbiome also communicates with the immune system via microbial metabolites such as short Euros chain fatty acids. Short Euros chain fatty acids act on immune cells, promoting anti-Euros inflammatory responses and maintaining tolerance. Together, these mechanisms create a dynamic environment where microbes help digest complex carbohydrates that human enzymes cannot break down. The resulting fermentation products provide energy for both the host and the microbial community itself. Disruptions in this balanced ecosystem can lead to impaired digestion, altered immune function, or increased intestinal permeability. Therefore, understanding the composition and diversity of gut microbes is essential for appreciating how they support normal gastrointestinal physiology. Pause 0.8. Dietary fibers enter the colon largely unchanged by human enzymes. Resident bacteria possess specialized polysaccharide-degrading enzymes that cleave these fibers into simple sugars. Fermentation of those sugars produces short-chain fatty acids Euros acetate, propionate, and butyrate Euros in a stepwise manner. Acetyl-CoA carboxylase first converts acetyl groups to malonyl intermediates before chain elongation. Propionibacterium species then condense two acetyl units into propionate via the methylmalonyl pathway. Butyrate emerges from butyryl-CoA formation, catalyzed by phosphotransbutyrylase and butyrate kinase in firmicutes. These SCFA diffuse across the mucus layer to reach colonocyte membranes. Butyrate binds GPR109A receptors, triggering anti-inflammatory signaling cascades within epithelial cells. Propionate is taken up by hepatocytes and converted into glucose through gluconeogenesis. Acetate circulates systemically, serving as a substrate for lipid synthesis in peripheral tissues. Collectively, SCFA production links dietary fiber intake to gut barrier integrity and metabolic homeostasis. Thus, microbial fermentation transforms indigestible fibers into bioactive metabolites that shape host physiology. Pause 0.8. Butyrate is a short Euros chain fatty acid produced by gut bacteria during fiber fermentation. Colonocytes, the cells lining the colon, rely on butyrate as their main fuel source. Inside each colonocyte mitochondrion, butyrate enters beta oxidation to generate acetyl-CoA. Acetyl-CoA feeds the citric acid cycle, producing NAD and FAD2 that power ATP synthase. The resulting ATP supplies energy for ion transport, cell proliferation, and barrier maintenance. When butyrate levels drop, colonocytes must switch to glucose or amino acids, less efficient pathways. Reduced ATP production can impair mucosal integrity, making the lining more permeable. This permeability allows luminal antigens to reach immune cells, potentially triggering inflammation. Thus, a steady butyrate supply is essential for sustaining epithelial health and preventing gut barrier dysfunction. Dietary fibers that feed microbial fermentation help maintain this critical energy flow. Pause 0.8. In the gut, microbial-associated molecular patterns are detected by pattern recognition receptors on dendritic cells. These receptors bind specific motifs such as lipopolysaccharide or peptidoglycan, initiating a signaling cascade that shapes immune tone. The activated dendritic cells migrate to mesenteric lymph nodes, where they present antigens to Na-tilde-VT cells in the context of co-stimulatory signals. Depending on cytokine cues, these T cells differentiate into distinct subsets, including regulatory T cells that produce anti-inflammatory mediators. Short-chain fatty acids produced by bacterial fermentation act directly on epithelial and immune cells through G-protein Euros-coupled receptors. Binding of butyrate or propionate to these receptors enhances the transcription of genes encoding interleukin-10, a key cytokine for tolerance. IL-10 then feeds back to dendritic cells, dampening pro-inflammatory pathways and promoting a balanced mucosal environment. The interplay between MAMP and SCFA thus creates a feedback loop that fine-tunes both innate and adaptive responses in the gut. This regulatory circuit prevents overactivation of immune cells, while allowing rapid defense against pathogenic invasion. Visualizing this process helps illustrate how microbial signals translate into cellular decisions within gut-associated lymphoid tissue. The diagram will show dendritic cell sampling, T-cell differentiation arrows, and cytokine signaling pathways highlighted in color. Understanding these mechanisms underscores the importance of a diverse microbiota for maintaining intestinal immune homeostasis. Pause 0.8. Microbial metabolites, especially short-chain fatty acids SCFAs, act as signaling molecules within the gut lumen. When SCFA reach intestinal epithelial cells, they bind to G protein-coupled receptors on the cell surface. This receptor engagement activates intracellular signaling cascades that culminate in transcription factor phosphorylation. Phosphorylated transcription factors translocate into the nucleus and promote the expression of tight junction proteins such as occludin and claudins. Elevated levels of these proteins strengthen the intercellular seal, reducing paracellular permeability. The enhanced barrier function limits the passage of luminal antigens that could otherwise trigger inflammatory responses. Thus, microbial SCFA indirectly preserve gut integrity by modulating gene expression in epithelial cells. Experimental studies show that diets rich in fermentable fibers increase SCFA production and upregulate occludin transcription. Conversely, reduced fiber intake diminishes SCFA availability, leading to lower tight junction protein synthesis and barrier compromise. These mechanistic insights underscore the importance of microbial metabolites in maintaining intestinal homeostasis. Pause 0.8. The vagus nerve serves as a bidirectional highway that carries signals between the gut and the brainstem. Microbes residing in the colon produce metabolites such as tryptophan derivatives that can travel along this neural route. These compounds influence the activity of enteroendocrine cells, which release serotonin precursors into the bloodstream. Serotonin produced in the gut can act locally on vagal afferents or enter systemic circulation to reach the brain. When serotonin binds to receptors on vagal neurons, it modulates firing rates that convey mood and stress signals. Simultaneously, microbial metabolites can cross the blood-brain barrier and directly affect neurotransmitter synthesis in cortical regions. This dual communication pathway illustrates how gut flora shape central nervous system activity through both neural and chemical means. Neural signals from the vagus nerve are integrated in the nucleus tractus solitarius, a key hub for autonomic regulation. The integration of microbial cues with autonomic output can influence sleep patterns, appetite, and even emotional resilience. Thus, the gut-brain axis exemplifies a sophisticated dialogue where microbes act as chemical translators for neural messages. Pause 0.8. A high-fiber diet supplies complex carbohydrates that many gut microbes ferment into short-chain fatty acids. These fermentation products, especially butyrate and propionate, serve as energy sources for colonocytes and modulate epithelial barrier integrity. Conversely, a low-fiber diet limits substrate availability, reducing SCFA output and favoring bacteria that metabolize host glycans. The shift in microbial community composition is reflected in bar graphs, showing increased abundance of firmicutes such as Faecalibacterium in high-fiber conditions. Low-fiber states often display higher relative levels of bacteroidetes and opportunistic taxa that produce branched-chain fatty acids. These metabolic changes influence host signaling pathways, including G protein-coupled receptors that sense SCFA and regulate immune tone. Thus, dietary fiber acts as a substrate that shapes microbial ecology, which in turn dictates the profile of metabolites delivered to the host. Visuals juxtapose these scenarios, allowing learners to see how nutrient input translates into measurable shifts in bacterial abundance. Bar charts illustrate the quantitative differences, while icons for SCFA levels emphasize the functional output of microbial fermentation. By focusing on mechanisms rather than outcomes, students gain a clear understanding of how diet orchestrates gut microbial dynamics. Pause 0.8. The gut microbiome is composed of diverse bacterial phyla that ferment dietary fibers into shorter chain fatty acids such as acetate, propionate, and butyrate. Butyrate serves as the primary energy source for colonocytes, sustain mucosal integrity. Microbial signals euros including SCFA and MAMP euros interact with guta euros associated lymphoid tissue to regulate innate and adaptive immunity. These metabolites also influence tight junction protein expression, preserving barrier function. Finally, microbial products communicate with the central nervous system via vagal afferents and neurotransmitter precursors, linking diet, microbiota, and overall health.